Okay, so today our objectives are to distinguish between an observational study and an experiment, and to explain the concept of confounding and how it limits the ability to make cause and effect conclusions. So, an observational study is when we observe individuals and measure variables without attempting to influence outcomes, the responses, the outcomes. Okay. An experiment, on the other hand, we do want to try to influence, we want to measure the responses in a very particular way. And so in an experiment, we deliberately impose treatments on individuals to measure their response. Okay. And we'll have more than one treatment. Oftentimes we'll have a placebo, a fake treatment, and we'll measure it against a treatment we think is working, just to have a control group. Okay. Now, if a experiment is done incorrectly, even if an observational study is done incorrectly, there can be confounding variables. Which confounding, it means confusing. It makes it hard for us to tell whether the explanatory variable is actually doing what we think it's doing. And we had that when we did our little experiment based on whether you could tell whether it was tap water or if it was bottled water. The confounding variable was the temperature. The temperature was colder in the tap water, and it influenced the decision that many of you made on whether or not one was tap water and one was bottled water. Now, so confounding, it occurs when two variables are associated in such a way that the effects on the response variable cannot be distinguished. Not a Q, that's an A. Cannot be distinguished. So let's look at an example here. Is soy good for you? The November 2009 issue of Nutrition Action discusses what current research tells us about the supposed benefits of soy. For a long time, scientists have believed that the soy foods in Asian diets explain the lower rates of breast cancer, prostate cancer, osteoporosis, and heart disease in places like China and Japan. However, when experiments were conducted, soy either had no effect or a very small effect on the health of the participants. For example, several different studies randomly assigned elderly women to either soy or placebo, so there's that idea of a placebo, and none of the studies showed that soy was more beneficial for preventing osteoporosis. So what explains the lower rates of osteoporosis in Asian cultures? We don't know. It could be due to genetics, other dietary factors, or any other difference between Asian cultures and non-Asian cultures, but we have a pretty good idea that's probably not soy from the experiment. And notice they compared soy to a placebo. They compared it to something, which is one of our basic things about the experiment. Okay? All right. And this one about coffee you can read through. Okay? And there's a link there if you want to look at that. All right, let's look at the next section here. So we can talk about the language of experiments. We want to be able to identify the experimental units, explanatory and response variables and treatments, explain the purpose of comparison, random assignment, control, and replication. Those are 4 very important things here. Right? And then describe a completely randomized design for an experiment. Right? So a treatment. It does not have to be a medicine or surgery or something like that. It is, a specific condition. In the case of, we're doing an experiment with trees, it might be, certain trees get so much, they get 2 gallons of water a day, and another group of trees get 3 gallons of water a day. Those would be separate treatments. It doesn't have to be medicine. Specific condition applied to individuals in an experiment. Okay. Experimental units are the smallest collection of individuals. A lot of times the experimental units are individuals, but not always. Okay. So the smallest collection of individuals to which treatments are applied. Okay. And a subject is just, it's when people are the experimental units. We don't like to call them experimental units when they're people. So we call them subjects, because that's much better, evidently. Alright, so let's look at an example here. A lousy situation. A study published in the New England Journal of Medicine, March 11, 2010, compared 2 medicines to treat head lice. An oral medication called ivermectin and a topical lotion containing malathion. Researchers studied 812 people in 376 households in 7 areas around the world. Of the 185 households randomly assigned to ivermectin, 171 were free from head lice after 2 weeks, compared with only 151 of the 191 households randomly assigned to malathion. So it says to identify the experimental units, the explanatory and response variables, and the treatments in this case. Okay, don't be confused. The experimental units in this case, the experimental units are not the people, they are the households. So let's say we're talking about the experimental units. It could be the smallest collection of individuals. Okay? And in this case, it was the households that were randomly assigned to each of the treatments. And the treatments, of course, were the ivermectin and the malathion. Now our explanatory variable and our response variable. Okay. The explanatory variable is the medicine given, right? The medicine given. And the response variable is the free from lice or not. That was the response variable. That's what we measured. The response variable is what's being measured. And we were Our explanatory variable was the medicine we gave them, and our response variable that we measured was whether the household was free from lice or not. Okay. Again, here's another one that you can do for yourself. Okay. Another example with growing tomatoes. Okay. Now let's talk about how to experiment badly. And we've seen a bad experiment because we did one. Okay. We want to make sure that we experiment well. Okay. So, of course, when I showed you my experiment with the water, bottled water versus tap water, I did not control all the variables. The tap water was refrigerated and the bottled water was not. Which helped explain the choices some of you made. You made decisions based on temperature, not on taste, and so there was a confounding variable. Okay? And we don't want to spend too much time talking about experimenting badly because we don't want you to do that. We want you to do it. We want you to experiment well. Okay? And there are 4 parts that we talk about when it comes to experimenting well. We've got 4 things: comparison, which is we compare 2 or more treatments. That's first. Okay, we have random assignment. So we use chance to assign the experimental units to the treatments. Okay, we want to create roughly equivalent groups by doing that. Very important idea. Create roughly equivalent groups. The third thing, okay, is control. Keep any other variables the same for all groups. Okay, and 4 is replication. And what that means is we want to be sure that the differences in our groups, if there is any, is due to the differences in our treatments and not because the groups are going to be different. The groups are always going to be different, and we will have some variation between our results, even between groups, even if the treatments were the same. Even if we gave everybody the same treatment in 2 different groups, there's going to be variation between the groups just because the groups can never be perfectly identical. So what we want to do is use enough experimental units so the differences in the effect of the treatments can be distinguished random variation in the groups. Okay. All right. Now, so let's talk about, we have one here where it says, how would you randomly assign 15 students to 2 treatments? We've done this before, so I will leave that to you if you want to do that again. Okay, we've done that. Let's talk about this one. Researchers in Canada performed an experiment with university students to examine the effects of in-class laptop use on student learning. All the participants in the study were asked to attend a university-style lecture and take notes with their laptops. Half the participants were assigned to complete other non-lecture related online tasks during the lecture. These tasks were meant to imitate typical student web browsing during classes. The remaining students simply took notes with their laptops. To assign the treatments, the researchers printed 40 papers with instructions, 20 with multitasking and 20 without, shuffled them, and handed them out at random to students in the classroom. At the end of the lecture, all participants took a comprehension test to measure how much they learned from it. The results? Students who were assigned to multitask did significantly worse than students who were not assigned to multitask. Explain how each of the 4 principles of experimental design were used in the multitasking study. Okay. So we have comparison. Well, certainly we compare multitasking to non-multitasking comprehension on a test. We certainly did comparison. Random assignment. Sure, they were randomly assigned. Units were randomly assigned. Okay, control. There's several ways that control was looked here. They controlled by the web. The lecture was controlled. The lecture was the same for all. The web browsing was simulated, so it was the same. And lastly, replication. 20 students in each group. Limits the effects of random variation. Good. All right. In the next video, we will talk about completely randomized design.